So now let's look at cell division. So this is a model for mitosis. We're going to look first at this cell. This cell in here, I could see the chromatin material, I could see the nucleolus, and I could see the nuclear envelope still intact. I could see all of the organelle around it. By seeing all of these structures, I know that this cell is in interphase. This cell here is also in interphase. Notice the nuclear envelope is still intact and the nucleolus is still there. Although we can see some of the condensed material start to condense in the form of chromosomes, but still, because we have this nuclear envelope still intact, this is when we're going to call it the cell is still in interphase. Notice here, the nucleolus is getting smaller in size, and we have the squiggly line over here. These are the condensed form of the DNA in a dividing cell, and these are the chromosomes. As soon as we see those chromosomes, we can tell that this cell is in prophase. This is another cell still in prophase. Notice how the nuclear envelope is about to disintegrate, and all of these different squiggly lines, you can see visibly clearly the chromosomes, and this is why we can say that this is in prophase. This is another cell in prophase. You could see that nuclear envelope is disintegrating. You could see the two centriole already replicated, and they are in the opposite pole of the cell, and they are sending out from them the spindle fiber, which are going to be used by those chromosomes so that they can migrate to the opposite pole. So this is a cell in late prophase, this is a cell in early prophase, this is another cell in early prophase. So you only need to identify that these cells are all in prophase. Next, the chromosomes are going to align in the middle of the cell, forming something known as the equatorial plate, also known as the metaphase plate. This is a cell that is in metaphase. How do you know it is in metaphase? Notice how all the chromosomes are aligned in the middle of the cell. This is known as the equatorial plate, and now the cell is in metaphase. Now, notice here the the chromosomes are migrating into the opposite side of the cell. This is known as anaphase. As soon as they start separating away from each other, we call it anaphase. Notice in this stage over here, the chromosomes have reached their destination. They are very close into the centriole. This is a cell that is in telophase. Notice also here the cell cytoplasm is about to constrict. This is known as cleavage photo, and all of these lines that you see over here are depiction of the spindle fiber that are coming out from those centriole. If you look over here, this is a cell that is in late stage of telophase. Notice here, as soon as I see the division between the two cells, at that time I know that this is cytokinesis. So this is a cell in telophase. This is a cell in late stage of telophase, about to become two cells with the constriction of the cytoplasm. That's when I call it cytokinesis.